Hello, and a very warm welcome to everyone joining us for today's Select Science webinar titled Architecture and Stoichiometry of Human Shelterin using Single Particle EM and Mass Analysis. My name is Lawrence Howes and I'll be moderating today's presentation. I'm delighted to be joined by our guest speaker, Dr. John Zinder, Senior Scientist in the Structural Biology Group at Odyssey Therapeutics. This webinar will discuss the purification of Shelterin subcomplexes and reconstitution of the entire complex using full-length recombinant subunits. Following the presentation, we'll have time for a question and answer session. If you have any questions throughout the webinar, please feel free to submit these in the box to the right of your screen. So without further delay, I'd like to hand over to our speaker, John, and thank them again for presenting for us today.
Hi, I'm John Zinder, and today I'm going to talk to you about my postdoc work in TC DeLonge's lab studying the architecture of human Shelterin by single particle EM and mass analysis. So throughout the talk, I'm going to be giving visual guides. Up in this upper left-hand corner, you'll be able to refer to those as well as the title to sort of orient you, because we're going to be talking about a lot of different complexes and a bunch of different components coming and going. So if you ever get a little bit lost, just look to the top of the slides. So first, I'm going to give a little bit about me. So I've been in biomedical research for about 15 years, and my primary focus has been on characterization of protein nucleic acid complexes. I consider myself a structural and chemical biologist, by which I mean that I have expertise in the purification of protein and nucleic acids, as well as their chemical manipulation. And then I have a whole suite of structural biology and biochemical and biophysical assay expertise as well. So after getting my master's at the University of Colorado at Boulder, I did my PhD with Chris Lima at the Sloan Kettering Institute, followed by a postdoctoral fellowship at the Rockefeller University in the lab of TC DeLonge, where I'm going to be talking about this work today. So there we worked in close collaboration with Tom Waltz to structurally characterize the human Shelterin complex and its associated factors. Presently, I am a senior scientist at the Structural Biology Group in Odyssey Therapeutics in Boston, and I'm not going to be talking about any of the work that I'm doing there today, but hopefully sometime in the future. Okay, so let's start at the end. The telomere is the end of a linear chromosome. So in humans, it consists of many kilobases of double-stranded DNA with this TTA-GGG sequence repeated over and over and over again. This double-stranded region is followed by a 3' single-stranded overhang of the same G-rich sequence. And this sequence is specifically recognized by the Shelterin complex, which is 6 different proteins that I'm going to go into more detail about later. But it also is elongated by this Telomerase complex, which has an RNA component that encodes the telomeric sequence, or the reverse complement of this G-rich sequence, and then a reverse transcriptase protein subunit that uses this activity to add DNA complementary to this RNA, thus extending the telomeres and preventing them from shortening with successive cell divisions. Additionally, telomeres can be formed into this T-loop structure, where this 3' overhang is tucked into the double-stranded region of the telomere. And Shelterin is intimately involved in both the recruitment and regulation of telomerase, as well as the formation of the T-loop. So it really sits at the center of a lot of the interesting biology and biochemistry of the telomere. So I'm going to elaborate on the end protection problem, which is what Shelterin solved. So this, simply stated, is that DNA damage, specifically double-stranded breaks, are terrible insults to the cell and need to be addressed before further cell division. And so this is accomplished by the DNA damage response, which will ultimately result in the repair of these double-stranded breaks or growth arrestor cell death. So the problem is that telomeres actually look like one half of this double-stranded DNA break. And so if the cell were to recognize them as damage, it would cause, the results would be catastrophic. And so Shelterin, in forming the T-loop and its other functions, is able to specifically repress the DNA damage response at the telomere. And that is how it solves the so-called end protection problem. So Shelterin is primarily composed of 2 double-stranded DNA binding proteins called TRF1 and TRF2. These have very similar domain architectures in that they have a extreme N-terminal charge domain followed by a dimerization domain called the TRFH domain, and then a long linker and a C-terminal MIB domain. And it's these MIB domains that actually recognize the double-stranded DNA of telomeric sequence. RAP1 binds to TRF2, and TIN2 simultaneously binds to TRF1, TRF2, and the TPP1 protein, which bridges it to a single-stranded DNA binding protein called POT1. So TIN2 is sort of thought of as the central organizer of Shelterin, insofar as it binds to both of the TRFs and bridges it to the single-stranded DNA binding activity. So decades of biochemical research have given us this sort of low resolution connectivity sort of map, but also structural work has figured out all the crystal structures and NMR structures of all the minimal folded domains within this complicated complex. So here we have the dimerization domain of both TRFs. We have that bound to the TIN domain of TIN2 to TRF1. And so on and so forth. But these are many, many different bits and pieces. I think it's about a dozen crystal structures or NMR structures in this particular slide. And so I like to borrow an analogy from Elena Conzi at this point, where what we have here are a bunch of Legos. So these are bits and pieces where we have rules as to how they can fit together, but we don't actually know what they build. And so what we have are these Legos, and what we want is to see what they actually look like when they come together in their fully functional form. And so when I came to the lab, there were protocols for making this complex. However, they were not optimal for my needs, for structural biology and biochemical needs. And so we were only able to get microgram scale with these protocols. And so I recloned them through a number of steps of optimization, only some of which I'm going to go into today. I was able to reclone these for co-expression in insect cells using the Big Back system from Jan Michael Peters and Brenda Schulman's labs. And I actually found the most successful strategy was to split them up into subcomplexes that I could then mix and reconstitute into the whole complex and purify by a sizing step. And so we were able to get milligram scale from these protocols, and that was enough for us to do sort of much more in-depth biochemical and structural studies. And so since I haven't gotten a chance to publish this before, I wanted to go at least a little bit into the technical challenges that I faced while trying to manipulate these complexes. And one of the most huge setbacks I faced was when I realized that there's actually an internal 3C protease cleavage site within the TPP1 subunit of this complex. So 3C protease is commercially marketed as precision, and so that might make you think that it's very precise. However, we were actually able to see in this experiment, I incubate with different combinations of proteases. So here just 3C, here TEV, and here both of them. And you can see in both of the conditions where I have 3C, you see degradation of the TPP1 band. However, if I only include TEV protease in the mixture, we don't see any cleavage of that. And so the solution to this was I had to reclone everything with TEV sites only. And then I also found that the TRF1 containing apo complexes stick to gel filtration resins. So I can show data later that shows that it's not just that they're insoluble, but if I have the complex without TRF1, you see this really nice peak this is a FSEC, so we're looking at GFP fluorescence, and the GFP is attached to TIN2 in this case. And so you see a really nice peak in the absence of TRF1, but as you titrate in TRF1, that peak sort of just absolutely gets squashed. And then if you give it a piece of DNA that it really likes to bind to, you see a much different pattern where the complex alone is sort of shifted to a nice monodispersed looking peak as you add in TRF1. And so the solution to this was basically that we only used glycerol gradients for sizing steps throughout the paper. I also noticed that TRF2 RAP1 displays aberrant behavior in EM, and this we still don't have a real good handle on. However, it was very reproducible. This complex that was monodispersed in gel filtration, active in functional assays, just looked absolutely terrible in every sort of EM experiment that we did. And we found actually that it this sort of aberrant behavior was not present when we studied it in complex with the other Shelterin components. And so we could only really do the structural studies of reconstituted complexes containing TRF2-RAP1. And finally, we also found that removal of the GFP tag, which we sort of retain on tin2 throughout most of the complexes, resulted in very poor behavior. And so this is the same complex where I've either cleaved or not cleaved the GFP tag. And although the GFP only accounts for about 10% of the molecular weight, you can see when we cleave it, we lose about 80% of the expected molecular weight product. And so it also looks much worse in the negative stain EM and in 2D class averages from that. So our solution to this was to retain the tag for all the structural and biochemical studies that we did. Okay. So on to I'm going to sort of fast forward a little bit into some more sort of unpublished results, and then I'm going to sort of use that to justify the methods that we used and the approach that we used in the paper that we ended up publishing. So this is a reconstituted Shelterin complex, totally uncrosslinked in negative stain electron microscopy. You can see nice discrete particles for each of the complexes. However, they're vastly different in their shapes and sizes. And so in order to solve this, we tried to use glutaraldehyde cross-linking, and more specifically graphics. So this is a technique in which, in addition to a glycerol gradient, you have a gradient of a fixative, in this case glutaraldehyde, which is a bifunctional amine reactive cross-linking molecule. And you apply your sample, in this case Shelterin, to the top of it. You spin it in ultracentrifuge, and as it's being separated by size in the glycerol gradient, it is experiencing at first very low concentrations of glutaraldehyde, which gently cross-links it, and then higher and higher concentrations of glutaraldehyde, which sort of stabilizes it and locks it in place. And through this sort of a protocol, we got these really nice sort of at least similar in shape at low resolution complexes from the same preparation. And we decided to go forward with this for Cryo-EM because we were noticing a bunch of denaturation during vitrification. And I'm sort of cutting a very long story short. This was not the only thing that we tried. We tried a number of different complexes, subcomplexes, DNA substrates, etc. But I'm sort of showing you one representative example where we got really nice contrast in Cryo-EM, at least in the raw micrographs. However, when we did any sort of data processing, 2D or 3D classification, we were not able to get it to converge on anything that in any way resembled any of the subunits in Shelterin. So these 2D classes, for those of you who are not familiar, at this particle number should probably start showing some proteinaceous features. We should be able to pick out, this is a TRFH domain, this is an OB fold, etc. However, none of these classes really look like anything resembling what we would expect the complex to look like. And so this was very disheartening because these were like cross-linked complexes. We thought that would surely help with this flexibility issue and lock it into one or several discrete complexes, but it seemed to totally just be in a random set of conformations. And we really wanted to get a handle on what this thing was doing in terms of its conformational flexibility, because this is sort of a negative result, and we wanted to have some way to actually physically observe this flexibility rather than just say, because the Cryo-EM didn't result in high resolution structures, it's too flexible. Okay. So for this, we turn to negative stain EM, and I can talk a little bit in the Q&A session if you are curious about why negative stain EM is especially good for flexible complexes, but suffice it to say, this was our reasoning for turning to negative stain EM. And we also decided that what we were going to do is, rather than just go for it with the whole complex, we were going to break the complex down into simple functional units. So not just like a single Lego, maybe like a cluster of Legos in that case, that we knew would start to inform on its function. And when we put these simple functional units together, we could sort of build up in complexity and see what is the turning point where we can and can't interpret the data that we get from EM. So the first functional unit we decided to look at, and the simplest, is POT1 TPP1 N-terminal domain, which has the full-length POT1 protein and then TPP1's OB fold and POT1 binding domains. So this complex has been well studied throughout the literature. We're not the first to look at it, obviously, and it purifies beautifully from the co-expression in insect cells. It behaves really nicely in functional assays. So here I'm showing an electrophoretic mobility shift assay where I have an N-labeled DNA probe and I titrate in the complex. And you can see at very low concentrations of complex, you get really nice binding to this probe. And if you mutate a single residue, you do not see that binding. Furthermore, it is specific to single-stranded telomeric sequence because it does not bind a double-stranded DNA probe. So everything's good there. It also looks really nice on negative stain EM. Although if you sort of zoom in on this, you can actually notice that no two particles really look alike. So we needed to come up with a way to sort of parse highly heterogeneous data in negative stain EM. And there's a number of different data processing pipelines and programs available for that. What ended up working for us was to use did our auto picking in EMAN, 2D classification in Relion to curate the particles and recenter them. And then we use that curated particle stack as an input for a 2D classification program called ISAC. And the output of that is shown here for POT1 TPP1. And this is kind of like a dizzying number of different conformations. But knowing a little bit about the structures of the individual parts of this complex, we were able to sort of come to some interpretations that made sense within these data. So I'm showing that here. Here are three crystal structures of the structured subsections of this complex. So these are the N-terminal OB folds, which bind single-stranded DNA of POT1. This is the C-terminal lobe, which binds to TPP1, and this is TPP1's OB fold. So these are the structured subunits within this subcomplex. And you can see in the 2D averages that you see features that very, very closely resemble these in terms of the relative size and shape. And so you have this bipartite lobe that looks kind of like two OB folds. You have an oblong lobe here that looks kind of like the C-terminal domain. And then you have a circular lobe here. But what you can also see is that they're sort of hinging relative to each other quite a bit. So the two lobes of POT1 are doing this sort of opening motion as I've depicted it here. And then the TPP1 OB fold is likewise flexing about its hinge relative to the POT1 C-terminal domains. And we've sort of cartooned our interpretation of this down here. So this is nice, but it's a little bit unsatisfying, and we really wanted to nail it down what these different domains were. And so we employed a trick where we have a 5' biotinylated oligo and we add Strep-DNA to it, purify that, and then do the same sort of analysis. And in this way, we can really clearly see the density consistent with the size and shape of Strep-DNA, which I've sort of put at the bottom of each of these 2D class averages. And then attached to that is a bipartite oblong lobe, which is the two OB folds, which bind to that DNA. And then attached to that and hinging sort of continuously again are those C-terminal domains of POT1. And you'll notice here that we no longer have the ability to resolve the TPP1 OB fold. We presume this is because the Strep-DNA and OB folds of POT1 are sort of dominating the alignment and blurring out POT1's or TPP1's OB fold. So at this point, we have a picture of this subcomplex where I've arbitrarily decided to make the C-terminal lobes of POT1 as the anchor non-mobile point and show mobility of the OB folds around this unit. And this basically showed us that, yes, these hinge domains between the structured subunits are allowing for pretty much continuous flexibility of the other structured subunits. Okay. Now we wanted to build up in complexity now that we understood that simple subunit. And so we added the rest of TPP1 and TIN2. And in addition to just doing the full-length proteins, we included mutants where the hinge domains were highly truncated, sort of hypothesizing that if we take a little bit of this flexibility out of the complex or potential for flexibility, we might be able to resolve discrete conformations a little bit better. And so we in this deleted construct, we have the hinge domain of POT1, which is 30 amino acids, replaced with 6 amino acid flexible linker. We have about 150 amino acids of TPP1's flexible linker replaced with this 6 amino acids, and we have a truncation of the flexible C terminus of tin2. And so these are the purified complexes, both the wild type and the triply truncated mutant. And so I'm sort of cutting to the chase here again. We could actually, going back to the POT1 TPP1 complexes, these are the sort of cartoon schematics and crystal structures that we used to interpret the 2D classes here. And we can see that there are related 2D classes from this complex further containing tin2, where we have another oblong density associated with the complex that we interpret as the tin2 TRFH domain. And so you can see this clearly resembles this class, but with this extra density. This class clearly resembles this class, but with an extra bit of density, and so on and so forth. So now the question was, what's the difference between this full-length complex and the triply truncated complex? And if you look at a couple of the classes from the full-length complex that are shown here, if you look at the triply truncated complex, it looks almost exactly the same. And I'm only showing a subset of the 2D classes. It has the exact same degree of heterogeneity and everything. And so these are our domain level interpretations of these 2D classes. And this was sort of a really surprising result to us, actually. Maybe it shouldn't have been in hindsight, but at the moment we were trying to rationalize this. And the model that we came up with is the toy snake model. And this is because I used to play with these sort of toy wooden snakes as a kid, and this sort of called to mind this. And in this analogy, the segments of the toy snake are like the structured subunits or the structured domains within this complex, and the flexible linkers are sort of extruding outwards from these. And you can see from this cartoon that even though these flexible linkers could be truncated to being very much shorter, you would still be able to get this flexing motion of these structured subunits relative to each other, even if you truncated these linkers. And so this gives us a sort of model like this, where we have these structured domains still flexing pretty much continuously. About, again, we've used the C-terminal domains as POT1 as an anchor. And we have tin2, its structured domain sort of associating differentially with that domain. I should also add that I'm not showing it here, but it's in the paper that this model is also supported by AlphaFold multimer analysis and by cross-linking mass spec that we did in collaboration with Yevgeny Nudler's lab at NYU and his senior researcher, Vlad Svedlov. Okay, so this is our understanding of how this slightly more built-up complex behaves. And now, finally, not finally, but next we wanted to actually look at how it would behave when it's bound to one of the TRFs. And so we chose TRF1 because TRF2 had already been sort of looked at by the Czech lab, and we thought that we might be able to show that TRF1, which is actually binds tighter to tin2 and is more directly responsible for bringing it to the telomere, we thought that it could actually form a fully dimeric complex. And so this is sort of a cartoon of it bound to the telomere down here. This is the SDS page. And there was an ongoing debate as to whether or not one tin2 would bind to it or two, because only one tin2 seemed to bind to TRF2, as shown by the Czech lab and CJ Lim. And so to get at this, we turned to the technique that is the reason I was asked to do this webinar, which is mass photometry. I won't go into the principle of it, but what really sold me on it was this paper published in 2020, where they showed that they had this sample that they had characterized by EM, and they were able to really clearly show the stoichiometry of the different classes that they were seeing in EM using mass photometry. And it was a single particle technique, which was also very appealing to me. And probably most importantly to me, it was entirely independent of conformation. So this particular complex they showed was very conformationally variable, and yet it showed a very sharp peak in the mass photometry experiment at the correct expected molecular weight of the complex. And so I thought that I might be able to use this technique in order to determine the stoichiometry of this, but also to get a measure of compositional heterogeneity within my sample. So this is sort of going back to an earlier criticism of the work from others, where if you see heterogeneity in EM, how do you know that it's conformational heterogeneity and not just like compositional heterogeneity? And so I teamed up. This technique was suggested to me by Tarun Kapoor, and I teamed up with his graduate student Lauren Vostal, who trained me on the instrument. And we had reconstitutions of this TRF1 complex and the absence of DNA and in the presence of DNA. Here I'm showing SDS page of the apo complex. And then these are fractions from a glycerol gradient with SDS page stained for protein and a native page stained for nucleic acid, showing that we have good coelution of protein and nucleic acid within this complex. And so I'm going to show you the raw data from mass photometry, which is one of the things I really liked about using this technique, which is you can right off the bat see that in the presence of DNA, you're only getting the same intensity of spot, whereas in the absence of DNA, you're getting all these different sizes of spots hitting the glass. And so when we quantify this in the histogram, we actually see that in the absence of DNA, we do have dissociation of this complex. We have quite a bit of heterogeneity. And so we have the full dimeric complex, or a species that is consistent with the molecular weight of the full dimeric complex. We have the same complex where one of these tin2 pot1 TPP1 modules has fallen off. And then finally, we have a complex where we have the thing that has fallen off from this complex ostensibly, and potentially we have another peak for TRF1 here. So it looks like things are sort of falling apart in the absence of DNA in this complex at least. And what was really satisfying is the whole thing coalesced into a single peak that was shifted by approximately the molecular weight of the DNA substrate that we were using in the presence of this DNA. So now we had all this nasty, heterogeneity in terms of composition, and DNA seemed to solve that entirely. I do apologize. I had a contaminant from my buffer that was giving me this low molecular weight species. I should have read the user manual a little bit more carefully as to how to prepare my buffers to get rid of this problem. Okay, so that was mass photometry to get at this. But this, we wanted to sort of take it further and figure out, chemically, do we have this sort of heterogeneity as well? And so we found that actually, if we lambda phosphatase, we found that our complexes were differentially phosphorylated, but if we lambda phosphatase treated them, they collapsed into single peaks of the expected molecular weight. And if we use this reconstituted phosphatase treated TRF1 complex, we were actually able to see this dimeric complex as well as it apparently this complex sort of falls apart during the mass spec experiment. And so these experiments were done by a really excellent senior researcher in Brian Scheidt's lab, Dom Telomeres, and we were able to see with pretty good accuracy, or to within error of the instrument, the dimeric peak in this technique as well. However, I'll emphasize that the native mass spec is a really difficult thing to do, so I couldn't run the experiment myself. It was done by Dom, and we needed quite a bit of material. So I think it was like 50 microliters of 5 micromolar material, which was pretty challenging to get at sufficient purity for the experiment. Okay, so now that we know that we're compositionally homogeneous, what does it look like structurally? And you probably guessed it by now, it was extremely heterogeneous, and so much so that we actually couldn't, sort of piece together the domain level architecture within these 2D classes. We wanted to try that streptavidin trick, so again, we employed DNA. This time, it's got 2 pot1 binding sites because this complex is a dimer. And we added the 5' biotin streptavidin moiety to it. And we just did this first with the most simple thing. And again, we can see really nicely the streptavidin, the 2 OB folds from each of the 2 pot1s binding to the substrate. And sometimes we could see the C-terminal lobes. But then once we add tin2, we basically lose all the features. We still see OBs in a couple of the classes, but everything else is blurred out. And finally, when we add TRF1, that really doesn't help our situation. If anything, it makes it more blurry, but it was really kind of nice and reassuring that we could see classes that sort of resembled other classes within this throughout all these different DNA substrates. And so the picture that we have now of the TRF1 complex is unfortunately not too clear, but it is a highly heterogeneous dimer. So each of these sides of this dimer are ostensibly moving independent of each other. TIN2 is connected to TRF1 via a long flexible linker again. And so we have these two POT1, TPP1, TIN2s things flexing freely about their flexible linkers, and all this flexibility is going on as well as these several conformations that TIN2 can adopt relative to the POT1 C-terminal domains. Okay, so finally we added TRF2 RAP1 to the mix, and in this case we can see the SDS page here, the purified complex. We do the mass photometry experiment, and we see that we have a peak that's consistent with the molecular weight of the dimer of this complex, as well as subcomplexes and that buffer contaminant down here. And then when we add a DNA that this complex should be able to bind, we see this dimer complex shift, but we still see these other complexes, subcomplexes as well. Perhaps the glycerol gradient wasn't really able to separate these so well, and there is some dissociation. So Shelterin again is a dimeric complex, and if we look at it in negative stain electron microscopy as we've done for the other complexes, again we see that the addition of TRF2 RAP1 does not decrease the heterogeneity. It if anything increases it, and the addition of DNA to that complex compacts the structure, but does not decrease the heterogeneity. Okay, so now we have this sort of cartoon level depiction of Shelterin along with its flexible linkers and this sort of idea of how everything can flex relative to each other. And we sort of asked, why would it be so incredibly flexible? And the answer that we came up with is obviously somewhat speculative, but we speculated that telomeres are actually dynamic and structurally diverse substrates. So it sort of makes sense to have a really conformationally flexible complex at the center of so much telomere biology. And by structurally diverse, I mean that they can be found in open states that are competent for elongation by telomerase. They can be found in T-loops where the 3' end is tucked into the double-stranded region. And all throughout the telomere, it is chromatinized. So it's all bound up by nucleosomes and nucleosome-associated proteins, chromatin-associated proteins. And so it sort of makes sense that you have a lot of flexibility within this complex to be able to accommodate all these really different sorts of structures that you have throughout the telomere. Furthermore, the telomere is a site of dynamic enzymatic processes. So as I've alluded to many times during this talk, the telomerase holoenzyme elongates the 3' end of it in the open state, and it has to be recruited to the telomere by Shelterin. The CST polyalpha primase complex is actually responsible for making sure that this 3' overhang is exactly the right size, and that's also recruited to the telomere by Shelterin. And then, of course, there is DNA replication happening in dividing cells. And this DNA replication actually turns out to be particularly difficult because of the G-rich telomeric sequence. And so Shelterin helps with this process by recruiting helicases to the telomere. So a lot of really interesting dynamic processes happening at the telomere, and Shelterin's sort of, again, at the center of everything. So I think I want to leave the talk with this idea, which is that there's maybe some hope for locking in some interesting structures of that full house of Legos that I alluded to before. And this comes from recent studies by my former colleague in the Long lab, Sarah Kai, and from a study from Kelly Nguyen's lab at the LMB. And I'll sort of focus on this at first because this is sort of hot off the press. It's just pre-printed right now. But what Sarah was able to do was she was able to get a structure of POT1 TPP1 bound to this CST complex, which is another telomeric. It's involved in that 3' end maturation that I mentioned before. And what's really cool about this is she actually was able to capture this in a state where these two structured domains of pot1 are sort of rigid relative to each other, or at least much more so in the absence of this complex. And in Kelly's work, they were able to resolve actually the two OB folds bound to DNA. This is a cross-linked structure, I should add, but they were able to resolve these two OB folds binding to the telomeric DNA that was being fed into the active site of telomerase, as well as the OB fold of TPP1 that was directly binding to the complex. And so these two structures sort of capture this functional submodule of Shelterin by stabilizing different parts of it in different discrete conformations. And so this kind of really supports this hypothesis that being able to access all these different conformations is important to the function of at least this structured module of Shelterin. And it also begs the question if we could get other telomere-associated complexes associated with Shelterin, could we unlock other discrete conformations that these full-length subunits can adopt? Okay, so with that, I would like to acknowledge my mentor, Titia DeLonga, and her lab, who really established a really wonderful place to work, our collaborators in the Cryo-EM core, who, although we were not able to get the structure to high resolution, I was very confident that it wasn't because of the microscopes, because they were in a very tight ship over there. Our collaborators in the Chite lab, specifically Dom Olaneris, who did the native MS work that I talked about, the Nudler lab, and Vlad Svetlov, who did the cross-linking mass spec that I didn't really have time to get into. And then here's a picture of the whole DeLonga lab. I also want to acknowledge Tom Waltz in his lab. I probably split my time, depending on the week, either 30-70 or 50-50 between the Waltz and DeLonga lab. And they were a wonderful bunch of colleagues to get to know and to work with. And our funding sources, of course, especially the Damon Runyon Cancer Research Foundation for funding such a risky project. I also wanted to leave with this. So this is actually the second meeting of the structural biology subgroup. Titia noticed at the first meeting that she was the only person without a beard, and her short-term solution to this problem is shown in this picture here. So I really like this, and should note that we later were able to find some non-bearded members to do some of this work. So with that, I'd be happy to take your questions. And if it takes you a while to think up a good question, or you don't feel comfortable doing it in a public setting, feel free to get in touch with me on Twitter. This is my handle, and I will probably be there for at least the short term. And I'd be happy to answer any of your questions if you reach out there.